Thank you very much, Nid. Thank you, Adam and Harry, for the invitation. I'm happy to disclose that I'm an employee of GSK, and the work presented here is the product of an amazing team of scientists and clinicians and our external collaborators. So, intrinsic to the concept of functional cure of hepatitis B virus is the notion that in order to achieve durable, long-term seroclearance of hepatitis B virus off all treatment, we have to achieve a competent immune system that can provide surveillance and destruction of cells producing hepatitis B virus. And so, so an important part of achieving functional cure is to address this immune dysfunction, which might be particularly peculiar to hepatitis B. And we've tried to simplify our understanding of hepatitis B immune dysfunction by separating it out into two components. A component driven by hepatitis B surface antigen level, and a component driven by immune exhaustion. Obviously, when we simplify things into two dimensions, we can lose some of the complexity and heterogeneity of the situation. And it's quite possible that for different patients with the different immune and virological phenotypes, for some people, the vector of these two components might be down here with a strong component driven by surface antigen, and for some it might be up here where you have a lot of immune exhaustion even for a low level of surface antigen. And then once you start to think about the diversity of the cellular and molecular processes that can influence surface antigen and immune exhaustion, then you realize that you're having to start to think beyond a two-dimensional graph into multi-dimensional complexity, which is going to require a lot of data and a lot of computational power to resolve. However, thinking about things in these two components is a good start. And we're fortunate that we've got Bepirovirsen, the antisense oligonucleotide, to use as a tool to start to tease out some of this complexity around mechanisms that could lead to functional cure, both in terms of control of surface antigen and methods to boost the immune response. So Bepi is very specific for hepatitis B RNA and binds specifically to it. And it then achieves destruction of those hepatitis B RNAs by recruiting a host enzyme, RNase H1. And RNase H1 is a cleanup enzyme. It's present in the cytoplasm and in the nucleus, and it's evolved to remove RNA that is stuck to DNA, because there shouldn't be RNA stuck to DNA. And consequently, it's able to achieve really deep lowering of levels of its target RNAs. The disadvantage is that in order to achieve that, it needs one-to-one binding of Bepi to the RNA. So if you've got a hepatocyte which contains a lot of copies of viral RNA, then you'll need a lot of copies of Bepi to be able to get that down to zero. Bepi has been shown to address the three mechanisms that contribute to hepatitis B persistence. It can reduce viral proteins by eliminating pregenomic RNA. It can reduce viral replication and viral DNA. And it's also been shown to have a moderate immune stimulatory effect, apparently acting as a weak TLR8 agonist. I think a lot of us are now familiar with the BeClear study, which was a phase 2 study looking at 4 different dosing regimes of Bepi in patients controlled on nucleoside analogues and in patients who were not on NA. What it showed was that if you look at the graph on the left-hand side, the green bars are the participants who had a surface antigen below the lower limit of quantification at baseline, at 12 and at 24 weeks of treatment, and 6 months after the end of treatment. I think these data emphasize 3 things. Firstly, that you see an increase in seroclearance during Bepi treatment, and then following Bepi treatment, you see that some of the people who serocleared relapse and others continue to remain hepatitis B surface antigen undetectable. Secondly, in the right-hand panel, you can see that as far as the primary outcome, which is being surface antigen and DNA undetectable, you see a greater response in those patients whose baseline surface antigen was below 3,000, and even higher in those whose baseline surface antigen was below 1,000. It's very difficult to get good epidemiological data to say what proportion of hepatitis B patients fit into those categories, but looking at the ones who entered our study, it's 50% or more who were in that 3,000 or lower group. And then the third thing is that looking at the different response rates in terms of surface antigen lowering between treatment groups, there is still some variability between groups, which is not fully accounted for by differences in baseline surface antigen. So what determines which patients respond to ASO therapy and who relapses? So we can start to investigate this, and this is from my colleague Amir Yusuf, who used pharmacological modeling to model out the surface antigen changes from the BClear data. And you can see that there are participants in grey who either don't drop surface antigen very much or don't get down to the lower limit of quantification, and none of them go on to become seroclearance patients. So undetectable surface antigen during Bepi treatment is necessary for somebody to become a seroclearance patient, but it's not sufficient. You can see the patients in blue whose model traces get down below the limit of quantification, but as we model that change out below where we could actually measure it with the assay, they don't get terribly low below the LLOQ and eventually come back up again and become relapsers. But then there's a third group whose surface antigen trajectories are modeled out in green, who get below the LLOQ and then continue to decline and eventually get down to a level where they cannot relapse. And from the model data, it looks like that threshold, which is necessary to achieve a sustained response, is around 10 to the minus 5 logs. We can then aggregate all those traces into a probability curve. So here you see the probability of achieving a sustained response 6 months after the end of Bepi treatment against the log to the base 10 of the hepatitis B surface antigen nadir value, so the lowest value achieved on treatment in the model data. And you can see that this divides the model participants into three zones. So there's this zone where there are the non-responders and the partial responders whose surface antigen never became undetectable. There's a middle zone where you have those who became undetectable and appear to be responders but then relapsed. And there's a final zone where the majority of participants, when they got below that 10 to the minus 5 threshold, became sustained responders, still negative 6 months after the end of treatment. We actually developed this model while we were already starting the Be Together study, which was a study which treated with 12 or 24 weeks of Bepi and then followed with 6 months of pegylated interferon. And as we began to look at the model data, we then began to think, okay, if we're hypothesizing that adding pegylated interferon after Bepi can improve the seroclearance rate, how might that mechanism be working? And there are three main possibilities. So the first is that we know that pegylated interferon can reduce surface antigen. So maybe sequential pegylated interferon might actually improve seroclearance by further reducing surface antigen. So it might move people from partial response into full response, and it might move people from being potential relapsers to being permanent responders. So it could have an effect of pushing people into that sustained level. Second possibility is that because pegylated interferon is increasing immune reactivity, it might actually have an effect on where the threshold lies. So maybe if you've boosted immunity, you no longer have to get surface antigen below 10 to the minus 5. That threshold might come up to 10 to the minus 3, or if we're really fortunate, up into a detectable level, so that we then get people who trigger a cure at a higher level of surface antigen. And then the third possibility is that by stimulating the immune system with pegylated interferon, we achieve a balance between surface antigen generation and immune response that holds some participants in that unquantifiable level in the middle. Maria Buti will be presenting the data from Be Together in the AASLD meeting. I'm not going to steal her thunder by outlining the results, but I think you can tell from the published abstract that the truth is likely to be somewhere in those second two options rather than the first. So from these data, it looks like achieving very low surface antigen is important for being able to achieve sustained seroclearance for hepatitis B. But we still see some heterogeneity in response. And this is understandable even thinking purely in terms of virological activity. You might imagine that if you have a small number of copies of viral RNA per hepatocyte, then a 300 milligram dose of Bepi should be able to eliminate all of those and achieve seroclearance. Whereas if you have a large number of copies of viral RNA per cell, then the same dose of Bepi might not be able to achieve seroclearance, even if that surface antigen is being produced by a smaller number of hepatocytes. So if we look to be able to increase the sustained seroclearance rate with Bepi, we need to think both about virological mechanisms and immunological ones. So how might we improve surface antigen lowering to improve seroclearance rates? So there are other strategies we can look at that might be complementary to the Bepi ASO. Firstly, PAPD5 and PAPD7 inhibitors can interfere with the host mechanisms by which hepatitis B stabilizes its messenger RNA. And so if we can reduce the stability of the RNA, we can reduce RNA and hopefully viral proteins by a second mechanism. We know that PAPD5/7 inhibitors have had toxicity challenges, which make them unlikely to be successful for long-term dosing. But if they were part of a short-term regime in combination or before or after Bepirovirsen, then maybe they've got the possibility to add some extra effect. We could, however, go farther. We know that small interfering RNAs have a very marked effect on lowering surface antigen. They work by a mechanism distinct from Bepi, by recruiting the risk complex, which is active in the cytoplasm. They break down the RNA, and by a method that it has evolved mostly to modulate gene expression. So that might explain why they tend to show a plateauing effect, getting surface antigen down very low, but not clearing very often. But perhaps if we can get the RNA down to a low level with a small interfering RNA, we then push more patients into a zone where Bepi can push them right the way down to that low seroclearance zone. Lastly, by sequencing with immune modulation, we've also got the opportunity to increase durability with either non-specific or specific strategies. We've already heard a lot about these, but potentially sequencing Bepi with a TLR agonist like TLR8 could produce additional non-specific immune activation. If PD1 is a specific component of immune exhaustion for a particular patient, then perhaps the PD1 and PDL1 strategies that have already been discussed might work more effectively in patients who've already had some surface antigen lowering. And then finally, Vensi's talked to you about targeted immunotherapy, similar to the therapeutic vaccines that Ellie spoke about. We know that many of these have been through development and have found it difficult to show a functional cure response, but maybe in somebody whose surface antigen has been reduced to a very low level by RNA interference mechanisms, then those targeted immunotherapies can have their greatest response. So in summary, functional cure of hepatitis B with Bepirovirsen appears to require achievement of a very low hepatitis B surface antigen nadir. So only those with relatively low baseline surface antigen have so far been observed to achieve seroclearance. Immune modulation with pegylated interferon might modify this relationship in a number of different ways, but importantly, pegylated interferon doesn't appear to be necessary to achieve Bepi-associated seroclearance. And sequential or combination use of a second surface antigen lowering mechanism or additional immune modulation should be investigated for its potential to make Bepi-associated functional cure a possibility for more people who are living with chronic HBV infection. Thank you very much.